So we're going to be open for questions at this point.
Hi.
Questions?
Patty Gans, UCLA. I had a question to our last speaker, Anne Marie. It just dawned on me that, the ability to annotate these specimens is very, very important, because it's not just, well, what do I need to do about the driver or the passenger, but really who got that cancer? Did they smoke? What other medications were they on? Perhaps their ethnicity, their gender. I mean, a whole bunch of, as a population scientist, that's what I think about, because we'd like to prevent those advanced cancers and know something about the context in which the mutations occur. In addition, Roy did a nice paper a couple of years ago in JCO about the use of tobacco that continues in many patients who go on our clinical trials. So behaviors of the individuals who we're going to treat with these targeted therapies. So I think it's a very, I really admire what you're trying to do, and it's an enormous undertaking, but this is really to anyone who wants to answer this. I don't know what's happening in the lung map trial, but collecting additional data about the host in which the cancer occurs seems to me a missed opportunity if you're not going to do that.
So in the context of our clinical studies, we do collect on quite a lot of data on patient demography and particularly as it relates to lung cancer, for instance, we have information about their smoking history and other elements. Now, obviously, you're not going to be able to collect huge amounts of data, and I think the key element in the future will be the ability to tie the genomic data back to electronic health record data, where that you have much more information along the lines of what you're suggesting.
Maybe. We don't collect smoking history even in the medical record. So,
Yeah.
I mean,
that's one approach you might want to consider. Yeah.
So, I mean, I think we don't know the pathways by which certain mutations are expressed or not. And again, you're spending all this money on doing the sequencing with just a little bit more questionnaire data, if you will. The enrichment of the biological data would be profound. So,
thank you.
Yeah, that's a great question. I think the nice thing about the lung map is we do have pretty broad consent, so we will be able to do a great deal of discovery. But as far as the smoking goes, yes, we work very closely with a number of individuals, including Stephanie Land at the NCI, and we actually have we didn't put the full enchilada. There was a large 50-page smoking survey that they had offered to us. We felt that would be too complicated. We do have a 2 or 3 pager in there. And you're right, this is a great opportunity where almost all these patients are smokers. We can look at dose, we can look at time from cessation, continued smoking at the mutational profiles.
I should add that when we conducted not a genomic study, but in a clinical trial, we did a companion epidemiological survey, and the take rate of people answering that questionnaire was way over 95%. They love to do that, and this is information they realize is very important to collect, and you may not be able to get it retrospectively.
And Bruce?
So Bruce Johnson, Dana-Farber. I have a question for Dr. Martin, and it's an operational piece about how you plan to employ this. So at most of the academic institutions, they have next-gen platforms. So is your plan that after those institutions do their own analysis, they send them in, or do you plan to deploy machines to the different sites? And how are you going to handle the folks who make up, a lot of the institutions that refer in patients into your trials have already employed a system like that. So do you anticipate being duplicative, or do you think this is going to supersede it, or how do you think it's going to work?
I think it's going to be a hybrid of what you've just described, because we do acknowledge that there are a number of centers who are conducting this type of analysis. We've heard it today. And I think that whilst I don't know if David Litwack's still in the room, I personally think that especially in where we're dealing with very rare patient populations, where we find those patients are through the screening that is already occurring at those sites. And so we would confirm the mutation with the assay that we're developing. We wouldn't insist that patient could be enrolled based on the local test, but we would confirm it for the purposes of the diagnostic. But then the master screening network would also create a mechanism for centers that don't have the ability to run that kind of testing. Because, you know, we're looking at this on a global scale, so there are going to be many centers that wouldn't have or have the infrastructure to do that.
Are you going to keep the machines central, or do you anticipate deploying them to the different centers?
I think to be honest with you, Dr. Johnson, we haven't discussed the deployment to the different centers, and I will take that back to the group. Thank you.
Can I ask a question?
Well, let's
Okay, after.
Hi, Sean Tunis from CMTP. It's a question for Dr. Herbst. A couple questions about the lung map. One is whether there's any sort of, what's the impact of having the lung map protocol on companies that are developing products for non-small cell lung cancer that are, don't get into the protocol? In other words, is there any kind of unintended negative consequence outside if you're not part of that, whether access to patients or anything? And maybe that's speculative. But the second question is, now that, you've done a lot of or you and others have done a lot of the kind of trail breaking to kind of develop this model, is the next, master protocol going to be 50% easier to set up, or is it, is it sort of everyone's going to be just as hard?
Right, those are good questions. Of course, there's always a complexity in clinical trials development with a competitive environment and what else is happening. I would contest that, we actually will help others develop drugs because we're promoting profiling. In many ways, we're paying for the biopsy and the profile. While I'm hoping that this will approve 5 drugs the first round, it's unlikely that will be the case, and some of the arms will fall off, and patients will have their results, which could then be used to put them into earlier trials, much of what Lillian was showing. We still need to develop new drugs and new markers, so I don't think it'll be an issue. Certainly, if someone wanted to run a trial, a company in the second-line squamous space, it might be hard, but I would hope that they'd see that there are economies of scale and cost to work with us, and we're constantly talking with groups and trying to bring them in. I really see this as a modular operation. Right now, we're first getting going, but once we're screening as many as 1,000 patients a year, hopefully we can use this as a way to look at many drugs at one time. Your second question is actually one I'm glad you asked, because we are seeing many groups come to us and want to do similar things. And I'm very proud of that, the fact that the public-private partnership idea has moved on. There's an aligned trial, an assigned trial now in colon cancer that's developing, and I speak with their leaders quite often, trying to learn from our lessons, and there have been lots of lessons. It's not easy, working with the public, the private groups, bringing all the academics into one trial, but bringing cooperative groups together. But it is working, and I am hoping that this sort of cooperation and some of the financial issues is how we've worked it out will help other groups, and I think it is. So there's a breast cancer group now. There's also, I didn't mention it, but out of our think tank, again, with a lot of force from the FDA to encourage us to do this, there's also the idea, what if you compared drugs of the same class all at once? Why do we have to do 10 ALK trials? But why don't we screen those 5% of patients for the ALK rearrangement and compare drugs head to head? And of course, the initial reaction was, oh, how could we do this? Companies won't participate. But through the NCI mechanism, this is moving forward, again, building on some of the spirit and some of the lessons learned.
Question?
Hi, Jennifer Malin, WellPoint. So thank you all for the presentations this afternoon. They were all terrific. And the question I have, so Roy, in your study, it seems like the sequencing is covered as part of the study. Patients have to enroll to get the sequencing done. And it sounds like with the GSK proposal and with your example with the BRAF study, those were done outside the trial and the patients came to you already sequenced or knowing that they had the BRAF mutation. I know the Novartis trial, signature trial, basically states that practices have to adopt next generation sequencing across the board, and then if they meet the criteria for the trial, they'll enroll them. So as a payer, and following up on Patty's great question as well, I struggle to see how we're going to really it seems to me like there's an implicit, well, it should just all get paid for, even though we don't know the clinical utility yet. And I struggle to see how we're going to annotate the specimens and have all the data if all this screening is happening outside the clinical trials, and then practitioners and patients are left to figure out what to do with the results.
Just to sort of clarify, I mean, the majority of the patients enrolled on the phase 2 study are ex-US, and in a large proportion of those patients are in France. And I don't know if you're familiar, but in France, INCA is paying for screening of all cancer patients, all the sort of main cancer types, but cancer patients for a panel of biomarkers that includes BRAF. So all lung cancer patients automatically will get screened for that. So we are benefiting from other programs that are screening patients for a series of biomarkers. However, in order for us to get any approval of our drug, we have to still demonstrate that the BRAF patient who is on the study is truly a BRAF patient using a validated assay, and have a mechanism in which to demonstrate that, as well as have an entire bridging study. So there's still the evidence we have to do from that perspective.
So you said it's INC in France?
INCA. It's the basically the National Cancer Group in France.
So it's essentially the National Cancer Institute has established a nationwide registrational study.
Yeah.
To answer your question, I think what you're saying is that people are all different, doing it in different sort of silos, and how we're going to actually learn from your data and my data and somebody else's data. How do we actually collapse this into one database? I mean, part of this, I'm sure Rich might talk a little bit about, cancer learning systems like CancerLink, where we would put all that data together. I hope that whoever's sequencing their patients in their institution are not just haphazardly not collecting it, and putting into some sort of meaningful database that ultimately you can use it to learn together.
Right. I agree, and actually, it's a very timely question. I just had a call about this today. So on the lung map trial, you have to get the sequencing done as part of the trial, which is good because we have it all linked. And we actually are saving tissue for bridging studies because we realize the foundation test might not be the ultimate test. But very often, since it's a popular test in the United States at least, people already have that test done. So they come wanting to do the trial, and they already have the identical test. They're using the identical platform. It comes off with a different piece of paper, but it's exactly the same. And so far, we're not able to use that. And it does hurt accrual a little bit, and it does seem a little bit inconsistent, but it's just one of the things we're having to work through.
Well, and I think the other part in terms of, I was at a meeting last week that NCCS hosted on cost and transparency. Given that all the payers in the U.S. say that they don't provide coverage yet for these trials, for these studies, because there's no clinical utility. So are these claims just getting submitted to the insurance company, and if they don't happen to pay, then the patient's liable for it? I mean, what's the informed consent process regarding the cost of these tests if they're being done outside a clinical trial context and there's no coverage, and yet companies like Novartis are essentially requiring them to make people eligible for a trial?
I'll just tell you, I think our institution eats a lot of the cost. The academic institutions are not billing for a lot of these.
Lillian, you wanted to have a question.
Yeah, I guess I have a question for Roy. Do you think it's ever realistic to actually do a trial like LungMap and get the strategy being an approval strategy rather than the arms being an approval strategy? In other words, the whole act of stratifying patients by their genotype and matching them will lead you to, an approval process. Maybe it's an unfair question to put you, because, I mean, ultimately, these small groups are still not really obviously power. You still need to do, for example, a follow-up study. Do you think this is too far-fetched to actually think for cancers where you don't have a lot of patients, the matching strategy itself is an approvable pathway?
Right, so to match or among multiple trials or multiple sequences, I think it's reasonable for more very rare diseases. Again, this paradigm I think is actually if we could just get this to work, where we're using one platform of next generation sequencing, and have that approved as our diagnostic, which has never been done, and to match it with different arms, working with multiple companies, I think that would be a good first step. But I see what you're saying, could you combine 2 or 3 efforts, one in Canada perhaps, one in the United States, and pool them together? So it's worth looking at.
Yeah, I guess I'm just worried that, how many umbrellas can we do in the world? And how much resources do we need to pay for all these umbrellas and baskets? And ultimately, do we actually have enough money and resources and patience to do an umbrella for every question?
Yeah, I think that's a good idea, but I have a. Well, yeah, we don't have enough money, right? We know that.
It's a big puddle.
Oh, I wonder for Lillian and for Anne Marie as well, have you thought about what you do if there is a situation where you need a tiebreaker? Let's say you have an SNV and you have a translocation. How do you assign the patient? If
That's a great question. So I will add that in the first instance, we're sort of cheating a little bit because these trials are pretty much completed, and so it's a retrospective validation of the assay, and anything additional that we find in the patient tumors, we will look to determine whether or not that has association with greater or less efficacy. So it's more of an exploratory approach, but moving forward, I think that's a challenge, and I think the NCI match design has come up with a good strategy to try and address that. But I think as we learn more about the coincidental nature of mutations and what that means for drug efficacy, that will help us to determine how best to adjudicate that. But I'd like to hear from the physicians on the panel, please.
I mean, I guess the correct answer is probably depends on which one is more functional and which one is more driver. But currently, the honest answer is in the phase 1 arena is an opportunistic. Whichever I have a drug for that actually would be a most reasonable match, that's what we would go for.
Yeah, it's a, again, you have to start somewhere, but, when you lump a number of different abnormalities together, it's hard to know. One of the thoughts I have from the master protocol is, again, with a very high bar from phase 2 to phase 3, of course, we picked the drugs in a way where we thought we would have the best chance of going forward. But if we don't make it past phase 2, then those data become discovery data. And we'll look at all the mutations, and all we need is a couple of responders, and we can work backwards, and that's the hope.
So I'll ask a setup question for future session in a little bit. Those who are doing somatic sequencing only, have you had much occasion to suspect that there's also a germline problem, and what do you do about it if you do?
In our current effort because we do collect blood samples for germline, so we have had situations where we have detected incidental germline findings, and we knew that was going to happen. So we actually have engaged our genetic counselors and actually have a fairly sophisticated way to, including in our consent, that patients will let us know whether they want to be informed, and if they do, we would have our genetic counselor in touch with them to give them the counseling, because we're not trained to do, and I'm sure Pat will touch on this tomorrow as well.
Is that going to play across the country?
Yeah, so we're working with, which is the branch of the NIH we met with? NHGRI. So all these become issues for further study. So what do patients do with the information? How do you return it? And it was brought to my attention, I didn't realize this initially, that even though we're only doing the somatic and sequencing tumor, we can figure out some germline abnormalities from the foundation profile. So these are things that we're looking into now, and we're going to actually add some sub-studies on. And we actually have some pilot funding that's being made available for that.
And when we have encountered that, Barbara, we have had to go back to the IRBs. And we have a process that we follow that notifies the IRB, and then we then go back to the investigator where the patient was enrolled to notify the investigator, so that there is a process that they will follow if they need to notify the patient.
If there are no further questions, I think our session is over. Thank you. Thank you.